and today it's my pleasure to introduce Dr. Lauren Lucas. Lauren and I actually met a couple years ago at a very small conference in Colorado where we bonded over our love for bile acids and rock climbing. At the time, she was wrapping up her PhD, which included her work as first author on one of the papers that originally identified these bacterial bile acid conjugates that turned out to have been really pivotal for the expansion and advancement in bile acid field that we've seen in the past couple of years. Now, Dr. Lucas works at University of Wisconsin-Madison under Federico Rey, where she's continued her work in bacterial bile acid metabolism, and this work has landed her a fully funded F-32 this year, which I think we can all appreciate. Quite an achievement. So with that, I'll hand it over to Lauren, and I'd also like to present her a plaque from.
Thank you so much.
Thank you for being here.
Great. Yeah, thank you, Erin, for that lovely introduction. Yeah, so I'm in my third year as a postdoc in Federico Rey's lab at University of Wisconsin-Madison, and I feel very lucky that I've been able to work on the bile acid system for so long. I did my graduate studies in bacterial bile acid metabolism and have continued that work in my postdoc. So what I'll be telling you about today is kind of like everything that I've done so far. And the title of my talk is Investigation of Bile Acid Transformations by Human Gut Bacteria Reveals Novel Activity.
Click on your slide. Click on your slide on your computer.
Sorry, what?
Click on this.
This light.
No, your computer.
Okay.
You probably are working.
Okay, the slide. Okay. Some background on bile acids. So bile acids are amphipathic molecules made from cholesterol in the liver. They maintain this four-ring structure. And there are two primary bile acids made by humans. This is cholic acid and chenodeoxycholic acid. And they get conjugated to glycine or taurine, also in the, oh gosh. Sorry, bear with me while I figure this pointer out. They're made in the liver and then stored in the gallbladder along with other components of bile until after a meal when they are released into the intestines. So these four conjugated primary bile acids travel through the intestines, and as they interact with bacteria, they can both be deconjugated and transformed. I'll tell you more about the pathway in a second. But then bile acids can either be excreted in feces or reabsorbed and circulated back to the liver in a process called enterohepatic circulation. Importantly, bile acids also spill over into systemic circulation and move throughout the body and can interact with different organs and receptors that way. And the bile acid pool might be something you hear me say, and people have different definitions of this term, but generally I'll mean the collection of bile acids given system at any given time. And to get into some more specifics, this is the canonical bile acid transformation pathway. This is kind of like a pre-2020 pathway where conjugated primary bile acids, the ones that I just told you about. Also, I have a little cheat sheet for some of the terms that I'll be using consistently throughout the presentation. So these post-derived conjugated primary bile acids, in this example, I'm showing glycochenodeoxycholic acid, right? So this is a chenodeoxycholic acid with a glycine conjugated at the C24 carbon position. These must be deconjugated as a first step to release free primary bile acids. So in this case, I'm showing chenodeoxycholic acid. And the enzyme responsible for this deconjugation is bile salt hydrolase, and these are bacterial enzymes. After bile acids are deconjugated, they can be transformed. One example is through the bile acid inducible operon, which removes the hydroxyl group at this C7 position. But what I'll focus on more today in this example is the activity by a 7-alpha hydroxysteroid dehydrogenase, which will oxidize the hydroxyl group at the C7 position, forming 7-oxolithocholic acid, and that's a secondary bile acid. So I'll be using this kind of notation throughout the presentation, so hopefully it'll become familiar very soon. And if you take nothing else from this presentation, I hope you can appreciate that these 4 conjugated primary bile acids that are host-derived through bacterial transformation become hundreds or thousands of different structures of bile acids. And these structures are super important because they all have slightly different functions and slightly different abilities to perform the functions that they do in our bodies. So the original function of bile acids that people discovered is in aiding absorption of dietary fats. So I mentioned that bile acids are amphipathic molecules. They can form micelles and help you absorb fat from your diet. But also I mentioned they circulate systemically throughout the body, and we have different receptors for these bile acids in every organ in the body. And through interaction with these receptors, bile acids can change not only your bile acid homeostasis, as you might expect, but also your cholesterol levels, your energy expenditure, inflammation, your insulin sensitivity. It seems we keep finding more and more ways that bile acids play roles in your body. And then finally, to add another layer to this really dynamic system already, is that bile acids have differential levels of toxicity towards your gut bacteria. So gut bacteria are transforming bile acids, but these transformed bile acids can also modulate the composition of the gut microbiota itself. And so it's no surprise that bile acids have been identified as biomarkers in therapeutics in many different systems in the body, through metabolic disease like type 2 diabetes, but also with liver disease, mast cell MASH, IBD, and various cancers of the gastrointestinal tract. And you maybe have even heard of bile acids as a way to exclude Clostridioides difficile infection, and even some parasites. The cessation is impacted by a presence of bile acids. And so because of their many different roles in the body and the health industry, the bile acid market globally is insane. In 2025, bile acid sales revenue was $363 million, with North American sales making up over a quarter of that. And the fields that make up most of this revenue is in pharmaceuticals, of course, like we just discussed. I didn't mention before that because of the bile acid nature, they are actually even be used as a drug delivery system, a targeted drug delivery system where they can bring, drugs to specific cancer sites, for example. Interestingly, they're also used in cosmetics because they are, apparently they help with skin elasticity, but they can also penetrate the skin barrier. And they are attractive molecules because they are naturally occurring. So I thought that was kind of fun when I was learning about this. And of course, in research, anyone who's purchased bile acids knows that they range from $50 a gram to $600 for 10 milligrams of bile acids. And just to add to this point, if you wanted to buy this report, you can get it for the low, low price of almost $5,000. Or if you're a student, a little bit cheaper. But if we want to reliably predict and modify bile acids to benefit human health, we have to, or I wanted to start by asking some very simple questions, right? Which bacteria can transform bile acids? What bile acid transformations can they perform? And how do communities of bacteria work together to transform these bile acids? In my graduate lab and the lab I still work very closely with, Daniel Amador Noguez, we do targeted mass spectrometry, which is great for identifying both known and novel bile acids. And then we can do targeted fragmentation to confirm structures of these bile acids. And we also do a lot of culturing, both in vitro and in vivo. And I'm so grateful to have access to a germ-free mouse facility at UW Madison, where we can introduce specific strains into the system and ask very directed questions. So I'm going to tell you about 2 stories today. In the first one, we identified, as Erin mentioned, kind of a novel class of bile acids, and there's a QR code for that paper that was published in 2021. And the second story, we identified a novel transformation pathway. This is the QR code for a preprint. We're currently in review at Nature Communications right now. But with both of these stories, you know, when you publish a paper, maybe this is more for the grad students, when you publish a paper, it's all nicely wrapped and tied with a bow, but science often doesn't follow that linear pathway. And so I'm going to kind of tell you the more narrative story today of how we came to these discoveries. So we'll start with the first story. I mentioned already that I'm interested in understanding what bacteria could transform what bile acids, but in this story, we're also interested in understanding how long it takes for these transformations to occur, as well as how keeping bile acids separately or combined affects their bacterial bile acid transformations. And so in this first set of experiments, I will include this pathway down here at the bottom, and that will tell you that I provided primary bile acids, so these unconjugated bile acids as the substrate. In this experiment, we used 72 gut bacterial species from our lab collection. These largely overlap with the human gut microbiome collection. Pretty fast growing, easy to grow organisms, which is great for us because we need them all in the same media to be able to run our samples and collect data quickly. So like I said, in this experiment, we have primary bile acids. I told you folic acid and chenodeoxycholic acid, abbreviated here as CA and CDCA, are the two human primaries. We also included DCA because it is the most common secondary bile acid that we see in humans. And we had each bile acid at 100 micromolar when it's alone, or if we had all three pooled together, it totaled 300 micromolar. And these are physiologically relevant concentrations that bacteria might experience along the length of the gut. And we sampled at both 24 and 48 hours. And so it looked a little bit like this, right? You have a tube of bacteria grown with media with bile acids in it. At certain time points, you can take we took a mill out, spun out the cells, did a dilution, and ran it on the mass spec to get our data. And so we run standards alongside our samples, which allow us to identify and quantify bile acids in our samples. And so just to orient you on how to read this heat map, I have the bacterial species of the 72 that were capable of bile acid transformations here on the left, with their phyla denoted by the colored dashed lines. I don't expect you to be able to read all of these in this time, but you can follow some patterns. And then here on the right, we have the scale bar. So boxes that are in the pinks or light blues are lower concentration, whereas dark blue is higher concentration. And this is for our 24-hour time point. Because we had bile acids separated, we could see, you know, which bile acid gets transformed into what across the top. And we have a second time point for 48 hours. And what we learned from this experiment was that in our set of species and our environmental conditions is that hydroxysteroid dehydrogenases were the most active enzymes. And they were most active on the C7 hydroxyl group, like I showed in my example of bile acid transformation pathways. In addition, we saw that most transformations occurred within 24 hours with just a little bit higher concentrations of transformed bile acids by 48 hours. And I'm not showing the data here, but it didn't really make a difference whether the bile acids were alone or mixed. These patterns were consistent across the experiments. But I told you I provided primary unconjugated bile acids in the media. So you can imagine my surprise and shock and fear as a graduate student when I started seeing conjugated bile acids in my samples. I was seeing glycochenodeoxycholic acid, glycolic acid, things like that where we have standards for, so we know what their masses and retention times are, of course, in our standards. And then when I was seeing those in my samples, I emailed a few different people and was like, what did I do wrong? Why am I seeing conjugated bile acids in my samples? And everyone that I talked to was like, yeah, we've never seen that before, so good luck. And luckily enough, as I was working through this data, another paper was published on microbially conjugated bile acids. So this was from Robert Quinn while he was, I think, finished up with his postdoc at Thorstein lab. And he had identified microbially conjugated bile acids. This was cholic acid conjugated to phenylalanine, tyrosine, and methionine, I believe. And this was really exciting for us because we were able to say, okay, these conjugated bile acids are real, they're in our samples. And so what I did was calculate the masses for every primary and secondary bile acid in our sample, or in our compound list, with every single amino acid conjugated to it. So this is a list of 70 bile acids. And I looked in all of my samples and found that DCA, CDCA, and CA, as well as 7-oxo-LCA, were conjugated to glycine and 15 other amino acids in our samples. And so this heat map is based on relative abundance of these bile acids because they're not commercially available, so we couldn't buy them. But we did see that bacteria in the Firmicutes, Bacteroides, and Actinobacteria were the most, or were the bacteria capable of producing these microbially conjugated bile acids. So I published this paper in 2021 and was very excited to see in 2023 it be recognized as a top-cited paper. Yes, that was a very exciting email. And so, of course, with these bile acids, what we did to confirm their structures was fragmentation. Bile acids are great because they have this really consistent fragmentation pattern. In this example, methionine DCA, we lose the hydroxyl groups, which gives us a standard, or gives us a peak at this mass. And then if you lose when you lose the amino acid, we get a peak at this mass. And then, of course, we get a peak at the mass of the amino acid as well. So for every column in that heat map that I showed, I created 2 of these diagrams, and we did this for every single one. And those are all in the supplementary data of that paper. So now we take this canonical bile acid transformation pathway and add a few more arrows. So these MCBAs were present also, right? Our conjugated primary bile acids were in the media, but we didn't identify the enzyme responsible for this transformation. Of course, that was identified here, as well as at MSU, I believe, that bile salt hydrolase was the enzyme responsible for conjugating both primary and secondary bile acids to glycine, as well as other amino acids. And I think at this point, kind of made me question a lot of the terminology that we use to designate bile acids, right? If bacteria can reproduce these conjugated primary bile acids, then really, what is a primary bile acid if it's supposed to be host-derived? And so this was an increasing complexity of the bile acid transformation pathway, and is important to think about when we're designing synthetic communities with predicted bile acid pool production. Moving forward, I will be talking about experiments that provided conjugated primary bile acids instead of unconjugated, because we wanted to get a better sense of the entire bile acid transformation pathway. What really happened was that we tried to design a community with predicted secondary bile acid production, and it didn't work. And we found that this step maybe was what was missing. So we wanted to learn more about it. And in addition to understanding the entire pathway, we were also thinking about how transformation dynamics impacted the development of the bile acid pool. And we used both monocultures and co-cultures to test these processes. Okay, so I'll have a similar schematic down here, just identifying conjugated bile acids as the initial substrate. In this experiment, we used 77 gut bacteria, largely overlapping with the first set, but not exactly, of course. And in this set of experiments, we provided the 4 conjugated bile acids that are human primaries, as well as TDCA, again, because it's highly prevalent. And we pooled the bile acids all together for all of these experiments, but at 2 different concentrations that might mimic, closer to the gallbladder or further away along the lake of the intestine. So totaling 0.5 millimolar or 2.5 millimolar. Same kind of setup. We have our bacterial cultures with media and bile acids. We just grew them till stationary phase of this for this experiment because the time points didn't seem to make a huge difference with the endpoint analysis. And ran our samples on the mass spec, of course. And so the way to read this heat map is a little bit different than the other one. Because bile acids were pooled, I determined the concentration of all bile acids at the endpoint sample analysis. And so in this first column here, I have the conjugated bile acids that were provided in the media. So same thing on the right, we have a scale bar. You can see these up here at the top are about 100 micromolar, right? That's about here. So that means that these conjugated bile acids remained in the media throughout the time, the growth course. In contrast, there are sections like this that are pink, and that means all the conjugated bile acids had disappeared from the media by the time of sampling. In the middle column here, we have the unconjugated bile acids. So these would be produced from deconjugating activity. And you see, there are some darker blue spots. That's because, deconjugation of both TCA and GCA would produce cholic acid. And so there might be twice the concentration of these unconjugated primary bile acids. And then in the far right column, we have secondary bile acids. And so these were not produced as much as they were in the first study where we were providing unconjugated bile acids. But we still see in the Bacteroidota, there was phylum name change between the 2 papers. The Bacteroidota are still producing our 7-oxo bile acids like they did in our initial study. And we see some other more unique transformations that are consistent with the literature and our previous study. We can discern phylogenetic patterns from this behavior. Like I mentioned, the Pseudomonodota don't really transform or deconjugate or transform any bile acids, whereas the Bifidobacteria do all types or both types of deconjugation, taurine and glycine. And we did see that when deconjugation occurs, secondary bile acids were also produced. So pretty consistent with what the literature said at this point. Great. And I'm only showing the 100 micromolar concentration here, but we found it really interesting. It didn't matter if the bile acids were at 100 micromolar or 500 micromolar each. The relative percentages of the transformations were consistent. And that's still something that we are thinking about today. We also found that deconjugation activity and microbially conjugated bile acid production correlated. This is also consistent with the literature. So here on the left, we have this green bar. So what I did here is I added up all of the amount of deconjugation for each of these species. And we see that most of these bars are dark green. So most of the species that produce MCBAs also had high levels of deconjugation. So just generally active bile salt hydrolase / transferases. And we can also see some like interesting observations here. For example, Streptococcus inventarius and Coprococcus pomus, they produce a lot of diverse MCBAs. And consistent with our previous study, CDCA is the most common steroid core to be conjugated to amino acids. And there are specific amino acids that are most likely to be conjugated. However, with this type of analysis, there are some limitations. We did a single time point analysis, which allowed us to use very large cohorts of bacteria, of course, but we were losing a lot of information about how quickly these transformations were occurring, which can be super important in the context of like a community setting, right? We also didn't have a lot of information about how growth was affected by bile acids. And also HSDs are reversible enzymes. So it's possible we were losing some of these transformations at certain time points throughout our data. So moving forward, we selected a handful of strains and did time series analyses. And so this is where the monocultures and co-cultures come into play. Here's one example. So Coprococcus coma, as I mentioned already, produces a lot of MCBAs, and we do see that its bile salt hydrolase activity is quite high. For a lot of these graphs, I will try and display them exactly this way. So on the left here are the conjugated bile acids, and this is just all one experiment, only separated by the type of bile acids that are being presented in each graph. It's a lot of lines, but typically our conjugated bile acids are in these like purples and pinks. So the conjugated bile acids go down, pretty much disappearing from the media by 12 hours, and simultaneous, of course, we see the unconjugated bile acids show up. And this is just one example of 4 bacteria that we had that had really rapid and non-specific bile salt hydrolase activity. And in all 4 of these bacteria, we also saw MCBA production. What we didn't expect, and I think is really informative, is the transience of these MCBAs. And so this kind of might inform the function that they're performing for bacteria, potentially amino acid sequestration, because they are going up and then disappearing over time. In contrast, we had bile salt hydrolases that were much more specific. So in this case, we have Polyncella intestinalis. It deconjugated only the taurine conjugated bile acids, so the TCA, TCDCA, and TDCA. Non-vertical. These bile acids were disappeared from the media concurrently to the appearance of our unconjugated primary bile acids. And then what we also see that follows is these 3-oxo bile acid production. And another example of a different type of specificity was with Bacteroides ovatus, where we have different conjugated bile acids being deconjugated at different rates over time, with preference for some conjugated bile acids over others. And this impacted with the rates at which unconjugated primary and secondary bile acids appeared in the media. So we have this increase in our CDCA followed by an increase in 7-oxo LCA. So that is the pathway that we have that I've shown already. And so these are just 2 examples out of 4 that we also have where a specific or slow bile salt hydrolase did not produce any MCBAs, but produced primary and secondary bile acids instead. Finally, this is a part I find most exciting, as we started pairing bacterial organisms together. So I've told you about this pathway where deconjugation must occur before secondary transformations can occur. This is what the literature states. But another thing that papers will say is that sequential transformations can occur by 2 different bacteria in the same community. And that makes sense, but there's not actually a lot of data in the literature to support this claim. So we were interested in performing sequential transformations, but also because we were investigating bile salt hydrolase, we wanted to see how different rates of activity, different types of specificity with bile salt hydrolase impacted the secondary bile acid pool. And so what we did was we have a couple I'm only showing 2 again out of 4. So we have 2 different organisms here with different bile salt hydrolase activity, and we paired them each with Bacteroides theta iota omicron, which has really robust 7-alpha hydroxysteroid dehydrogenase. So we have species A doing the first step and species B doing the second step. And then of course we pair them together. And we grew these bacteria for 72 hours and gave them the same 5 conjugated bile acids at 100 micromolar each. So Bifidobacterium angulatum is an actinomycetoda and has a very rapid and unspecific bile salt hydrolase. The conjugated bile acids are deconjugated very quickly, and of course the unconjugated bile acids show up. So I guess importantly, the unconjugated bile acids are in the oranges, and then our secondary bile acids are blue and purple. B theta had this interesting pattern where mostly nothing happened. And then after 24 hours, GCDCA was disappearing from the media. We didn't really know what to do with that because when I looked for all my bile acids, we did not see any chenodeoxycholic acid or any 7-oxolithocholic acid showing up. The Y scale here is very small compared to 100 micromolar of substrate provided, right? So we kind of shoved this out of our mind for a little while and kept on plowing through with our experiments. And we were very excited when we paired these two bacteria together to see that, of course, the conjugated bile acids disappear. And just as we had expected, primary unconjugated bile acids were in the media. And as they went down, we have an increase of secondary bile acids appearing. So we have our bifidoangulatum doing this first step, followed by B theta doing the second step, and we can see each step of this pathway occurring. Clostridium symbiosum has a very different bile salt hydrolase than Bifidobacterium angulatum. It's very slow. So only after 24 hours did C. symbiosum deconjugate any bile acids, and it actually only deconjugated taurine conjugated bile acids. And so we see an increase in the unconjugated bile acids after 24 hours. B theta, same thing, kind of just like, well, this is reproducible at least, but I don't really know what it means. But when we pair them together, I saw this really interesting thing where conjugated bile acids are disappearing from the media a little bit earlier than they did when C. symbiosum was by itself. And we're starting to see secondary bile acids appear without any production of any intermediates here. And when I was putting this figure together, I realized this was the only co-culture where this was happening. And I started scratching my head and getting a lot more curious about this observation. Like I said, in every other case, we saw the primaries as an intermediate step. And so I went back into my data and saw that as GCDCA disappeared, we'll do this, G7-oxo-LCA appeared. And so if you can remember back to my 2021 paper, I said that I made a compound list where I determined the mass of every single primary and secondary bile acid that we had with every single amino acid that was available, and had just like this long list of hypothetical bile acids. I was able to just kind of look through those and see that glycine 7-oxo-LCA at the correct mass and expected retention time was in our samples. And if anyone else does mass spec data, this might mean more to you. The differences in retention times between bile acids, whether they're conjugated or unconjugated, is pretty consistent. And so you can be more confident that your bile acid is the correct identity when these retention time shifts are consistent, even when you don't have a commercially available standard to confirm that. Another piece of evidence to support our hypothesis that this was G7-oxo-LCA was not only the fact that B theta has a very robust HSD, but also that Casey Tirio's group published a paper about B theta actually last year, and they had cloned the 7-alpha HSD into E. coli and showed through a proxy analysis that it seemed to still have activity on GCDCA just like it did on CDCA. So I was thinking what's probably happening is that GCDCA is being directly oxidized into a secondary bile acid, bypassing that first required deconjugation step that's in the literature. And this is what made sense in the context of the Clostridium symbiotum co-culture as well. So the way I'm showing this here, right, we have our conjugated bile acids disappearing. And then what we actually saw was conjugated secondary bile acids appearing in the media, followed by deconjugation to release these secondary bile acids. And so the B theta HSD was acting on primary bile acids, in this case, producing actually more diverse conjugated secondary bile acids than it did on its own. So really interested in how the bacterial interaction there is changing the activity. And that following this conjugated secondary bile acid production, there was deconjugation occurring. And to confirm the identity of this conjugated secondary bile acid, we worked with a chemistry lab on campus, Helen Blackwell's lab, and they had their postdoc Clay helped us or synthesized the G7-oxo-LCA for us. They did NMR to confirm its structure, and then we also did fragmentation to confirm its structure using mass spec and matched the standard peak to our sample peaks, of course. And we ended up actually identifying 6 conjugated secondary bile acids. Here are just 2 examples of the fragmentation spectra that we did, but they were both glycine and taurine conjugated oxo bile acids at the C7 as well as the C3 position. I anticipate there are probably 12 conjugated 12 oxo bile acids as well, but we didn't get them in high enough concentrations in our samples to get clean spectra. So there are 2 potential pathways for the production of these conjugated secondary bile acids. I told you that we already know bile salt hydrolases can reconjugate bile acids to glycine as well as other amino acids. So it's definitely possible that what we were actually seeing was a conjugated bile acid being deconjugated, transformed, and reconjugated by bile salt hydrolase. The alternative option is what I was thinking, right? The simpler explanation is usually true, is that a conjugated bile acid is being directly oxidized into a conjugated secondary bile acid by HSDs. So this is the pathway we decided to test. And with the help of another postdoc in the Ray lab, Malikarjun Jalala, he created an HSD knockout of B theta. So with the wild type, of course, we see the same pattern where after 24 hours, GCDCA disappears, G7-oxo-LCA appears. But when you knock out the HSD, nothing happens. The conjugated bile acids remain intact. No unconjugated intermediates appear. So there's no CDCA in the media to support the possibility that bile salt hydrolase is playing a role in this transformation. And so now we have added yet another branch of bile acid transformations to the pathway. And so, as I've mentioned before, we do untargeted methods. So I went back through all of the data I've already shown and looked for conjugated secondary bile acids in our samples. And we see that they were there the whole time. In this is the same heat map as before. We added a few more columns for those 6 conjugated secondary bile acids that I was talking about. And they are both glycine and taurine oxo bile acids at the C3 and C7 position. And if you look down here, we're getting actually pretty significant concentrations in a few of them. And in some of these cases, deconjugation did occur. But in some of these cases, there was no evidence of deconjugation occurring. I also went back into our monoculture data because I guess reviewers, we submitted this paper in January and reviewers were like, you found an exception to the rule with B theta. I was like, I don't think that's the case. And what's actually happening, I mean, the data shows that HSDs are acting concurrently on both conjugated and unconjugated bile acids in our system. So here is the colon cell intestinalis time series that I showed you before, where we see unconjugated 3-oxos appear after the unconjugated primary bile acids, but we also saw conjugated secondary bile acids appearing at the same time. And this makes sense because colon cell intestinalis is preferentially deconjugating only the taurine conjugated bile acids, which is leaving a mixture of both unconjugated and conjugated bile acids in the media. And its HSD just didn't care, right? It was like, I will oxidize that the C3 hydroxyl group either way. In addition, right, there are more cases where deconjugation is not occurring at all, and these bacterial HSDs will directly oxidize conjugated bile acids. So here's another Bacteroides that doesn't produce any unconjugated bile acids. It only produces this conjugated secondary bile acid. Yet, we still didn't have strong evidence, or we didn't have any evidence of physiological relevance at this point. So we did a mouse experiment, right? You thinking, well, you already have the knockout strain, no big deal. You'll just add B theta wild type to your germ-free mice. But something I haven't mentioned yet is that mice have a completely different bile acid pool than humans do. And if you noticed, we are using a glycine conjugated chenodeoxyfolic acid is the substrate for B theta. So not only do mice not have chenodeoxyfolic acid, but they don't really have glycine conjugated bile acids at all. And so what we did, which worked, was that we put GCDCA into sterile water and we fed it to the mice. So after 24 hours of inoculation and pollination, we swapped out their water and they were drinking GCDCA. And then 48 hours later, we euthanized the mice and collected their feces, did an extraction, dilution, ran it on the mass spec. And we were able to see that GCDCA does build up in the system, and only with the B theta wild type do we see this G7-oxo LCA produced. And importantly, with the knockout, we don't see it with either strain, but especially the knockout, we again don't see any intermediates in the pathway that would suggest a role of a different enzyme in the production of these conjugated secondary bile acids. So, right, we started with this very linear bile acid transformation pathway. And now what I'm thinking of this more as a much more complicated bile acid transformation network. And perhaps it's no surprise that it's been very hard for scientists to manipulate this system with targeted bile acid production, because there are so many different ways that bile acids can be transformed by so many different bacteria all at the same time. And moving forward, kind of beyond understanding which bile acids or which bacteria can transform which bile acids, I'm really interested in understanding when and why these transformations can occur. So not only increasing the complexity of our communities and moving from both in vitro to in vivo with a complex community, but also manipulating the environmental conditions, such as the nutrient composition in the media, but also the pH can make a difference, as well as the bile acid composition, of course, and the community composition. And I'm also interested in understanding how both the bile acid pool shapes the composition of the community and vice versa. And I would like to thank, my lab, the Ray Lab, as well as Daniel, my PhD advisor, for all of their help and support through all of these experiments. I feel very grateful to have been able to stay working on the same system for so long, because it is a complex one. And with that, I'm happy to take any questions.
Okay, so first question from trainee. Shoot.
Yes.
So, thank you for your talk. It was really wonderful.
Oh, thank you.
I'm sort of curious do you think it's possible that the conjugated bile acids do a better job of dissolving fat than unconjugated ones, like ignoring bacteria completely?
Okay, yes.
I'm sort of wondering if maybe it's just a way of feeding bacteria amino acids, because it's just making
Sure.
amino acids more permeable to membranes.
Yeah. So my understanding, and maybe someone else in the room knows more, my understanding is that the amino acid conjugation makes the bile acid more hydrophilic, which would actually make it less capable of helping with dietary fat absorption or crossing membranes.
Gotcha.
Yeah.
But it's still better at crossing a membrane than just an amino acid by itself.
I actually don't know. I mean, as far as I know, conjugated bile acids need transporters to cross membranes, and I don't know about humus. I probably should know.
And then why would a bacterium want to spend energy oxidizing these things?
It's actually an electronic receptor. So it could be gaining energy.
Gotcha.
But there's not a lot of data about bacteria breaking down bile acids in anaerobic systems, except for deconjugating taurine, I believe. There are some bacteria that will remove the taurine to use that as a nutrient source.
Thank you for the talk. I had two questions actually. First one has to do with more like the toxicity and the community
Sure.
dynamics.
Yeah.
So you in your in vitro experiments, you gave it the primary bile acids and DCA at a very high concentration, and DCA is like one of the most toxic bile acids. So do you think that kind of like stress to the bacteria will cause like different kinetic shifts towards like deconjugation or deoxidation?
Yeah. I actually don't know that I believe there is a huge correlation between toxicity and activity. It looks from the preliminary data that we had, it seems that even when a bacteria is not growing at all, it will still perform these transformations in theory to detoxify the bile acids. That's what I think is happening. I didn't show some of the graphs where we see bile acids disappearing from the media over time. So I think that is because they're becoming intracellular and probably are building up toxicity, but how that impacts activity, we still don't know.
Okay, and thank you. For the second question, like I think your data is really compelling in terms of the novel transformation, but I did notice that you're using beta, which has BSH, if I remember correctly.
Yeah.
So is it possible that it's just like a really long sequential, at least in beta? Like, have you tested this with any organism that has HSD but no BSH?
Yeah, the literature says that B-theta has a bile salt hydrolase. We have knocked out the bile salt hydrolase and don't see any change in anything at all. I've also never seen our B-theta in any system deconjugate anything. So it's possible that we have a slightly different strain with a mutated bile salt hydrolase that's not doing anything in our system. If bile salt hydrolase was active at all, I would expect to see any of the potential deconjugated forms. We've tested toro-beta-myric-folic acid, toro-alpha-myric-folic acid, and I actually see nothing ever with this B-theta. I know that, Sloan Deflin's group shows that B-theta has activity on some conjugated bile acids. I know that Casey Theriot's group also shows this, but, not to knock anyone, but Casey Theriot's group doesn't do mass spec. They use like assays to interpret activity. And so potentially there's something going on there. I think for us, when I was designing these co-cultures, B-theta was our best candidate because it didn't have bile salt hydrolase activity actually. And it is quite challenging to find a bacteria that has secondary transformations without bile salt hydrolase activity. Yeah, good question though, and good catch. But I think TBD on the literature.
Yeah.
And like I said, environmental conditions do change activity, right? So we have moved from a richer media to a less rich media, and we see different transformations occur.
Oh, wow.
Yeah, so it could be the environmental conditions too.
Very exciting. Thank you. Yeah.
So just a general comment, Lauren's work has changed this bile acid field like overnight. I mean, I think that's an incredible achievement, I guess, as a graduate student, right? Or,
Yeah.
I mean, it's amazing. So like, we've all benefited from it. So it's just awesome work. Yeah. And then my question is, like, we now see beyond amino acids, like
Yeah.
polyamines, drugs, other things that are being conjugated to bile acids. So is it truly like it's like a transport thing, or it's a way to store other important metabolites, or is it just sloppy chemistry? I mean, that's what I'm curious about.
I doubt it's sloppy chemistry. I think, if Sita wrote the paper that was like, we think that these bile acid conjugates in picomolar concentrations are signaling molecules, and that would make sense for the different receptors that we have in every organ in the body, right? Yeah, and also, thinking of the different conjugations that occur. I think I want to probably increase this mass range that I'm looking at now because there are probably novel bile acids that we haven't discovered yet, of course. Their functions, you know, that's the hard part. And we were talking about too, like the system is so dynamic. It's so hard to nail down a specific function of any specific bile acid within the host context. I don't know though. I mean, the fact that the MCBAs are transient in nature, and like I'm pretty sure it was your paper that showed it's the amino acids that are produced by the bacteria that get conjugated, not ones that you supply necessarily. So it could be a source of sequestration of nutrients, saving them for later, trapping them in the gut, things like that.
Thank you so much. I was curious if you guys know why exactly the bile acids are like toxic to the bacteria when they build up. Is there like a specific reason why?
Yeah, so for B theta specifically, actually the GCDCA and CDCA disrupt its membrane. So it creates a leaky membrane. And so for some reason, oxidizing that 1-hydroxyl group solves the problem. And I think that makes sense for why GCDCA is also being transformed, right? It's like it's a detoxification mechanism. There aren't a lot of studies specifically looking at that. And historically, people will say, oh, one group of bile acids is more toxic to bacteria than another group of bile acids. But I think it's a lot more detailed than that. I think there are specific bile acids that are going to be toxic to specific bacteria, but these umbrella statements are not really holding as far as I can
So
tell.
probably wouldn't have much use as like a targeted treatment towards like dysbiosis of certain things.
It depends, right? So it depends what bile acid is associated with what health outcome and what transformation you are targeting. And so, you know, something I kind of, I don't know if I could go all the way back. Something I kind of talked about was that C7 position with dehydroxylation, right? Sorry, bear with me. That dehydroxylation will produce DCA, and we can just use this one. So this group, right, if it gets removed from an unconjugated bile acid, if it's CDCA, it produces LCA, and if it's folic acid, then it produces deoxyfolic acid. So these are toxic bile acids to both humans and gut bacteria. Something that I've been thinking about as a way to target that transformation pathway is through these HSDs. If you could divert the bile acids more towards these oxobile acids and away from DCA, LCA production, maybe you're detoxifying the bile acid pool in a sense. But these bile acids, right, some of them still might be toxic to your gut bacteria in ways that we don't yet.
Is it toxic to humans in the same way that it's toxic to bacteria in terms of like cell membrane disruption?
I believe the DCA and LCA are, yeah.